Hey everybody, welcome to my molecular biochemistry series. In this episode, we are talking about PCR, or polymerase chain reaction, which is a laboratory technique that allows us to amplify DNA or create more of them. And I have a memory device here called DATE that will allow us to remember the steps of PCR. And in order to do that, we're going to go on a date with somebody to amplify our relationship with them. The first thing we are going to do is turn up the temperature such that we denature our relationship a bit. We are going to get emotional, we're going to fight, and we are going to just raise the overall temperature. Then we're going to cool things off. We are going to amend things. We are going to anneal our relationship such that we are going to add in some primers to the relationship and allow us to reestablish our harmony and build more bond between us. Lastly, over time, we are actually going to elongate the relationship. We are going to make it even closer and more profound in our connection and our relationship. And this is the elongation phase, and it is the last phase, such that we now have many, many strands of DNA. It has been amplified just as our relationship has. In previous episodes, we talked about DNA replication in its natural environment, such that the cell needs to replicate its DNA during the S phase, which is prior to the M phase, in order to have DNA for when the cell divides during the M phase. So the new cell needs an instruction manual to tell the cell what proteins to make, to code for and instruct the cell to function properly and do what it needs to do in order to be a functioning living cell. So that's what it happens in nature, but we can manipulate this and analyze this and measure this using PCR. So the first thing we need to do is take a sample, such as a blood sample or saliva sample, or some way to gather the cell that we want to analyze. We need to lyse the membrane of that cell so we can extract the DNA. Once we've extracted the DNA, we need to eliminate any of the other components that might degrade the sample. So we can use a protease to break down all of the protein components that might be surrounding the DNA without denaturing it itself. Once we do that, we can place it in our sample, and then we can finally denature that DNA such that we break the hydrogen bonds between each strand, and so we now have two separate single-stranded DNA strands. Then we can begin the annealing phase, where we cool things off to roughly 55 degrees Celsius, and we can add in our primer, which is a specific primer to boost our relationship or boost the connection with a new DNA, such that polymerase can come in and add nucleotides to the strands. This allows for the third phase of elongation, where we can add the different nucleotides and complete the double helix, such that there's now two double helices, and we've now moved from 1 to 2, and we can repeat this process 25 to 30 times, such that we've increased many order of magnitude the number of DNA helices in our sample, billions, if not millions, if not billions of DNA helices. This allows us to analyze them, manipulate them, and measure them much more easily. Some applications, for example, would be we can draw blood, we can look at the leukocytes, the white blood cells in that blood. We can extract the DNA and amplify it such that we can identify if there are any cancer-causing genes in that DNA, such that we can early detection from possible tumor formation. Another option is if you're at a crime scene and forensic scientists can sample a tiny fragment of DNA, we can bring it to the lab, use PCR to amplify it, and then easily look at whose DNA is it. Another option is we could gather a piece of DNA, we could amplify it so that we can easily change a nucleotide here or there. We can manipulate it, forcibly mutate it, and then insert it back into another cell and see what output comes through. We could see if the outcome is what we want it to be. We have in fact played God with the cell. And so those are some of the applications, and that I think is a good breakdown of PCR. And so with that, I'll see you guys in the next one.